Well, so I'm going to attempt to take us from the periodic table to the structure of the ribosome in 15 minutes. So I apologize in advance if I don't achieve that. The bottom line is that life very likely started with RNA or something like RNA. RNA invented protein, and RNA invented DNA. And that's how we ended up with, let's see, where we are. So life is based on two languages, the language of nucleic acids and the language of proteins, as you know. And these are completely unrelated languages, as different as English and Chinese. And this is, as you all have seen, the central dogma. DNA encodes the genotype, and proteins, for the most part, create the phenotype. So how did this system evolve? And a possible scenario, as I said, starting with the RNA world, and then you have proteins, and you have a RNA protein world or RNP world, and then RNA DNA protein world. So the RNA world emerged from the discovery of ribosomes by Chak, Pace, Altman, and others, and now there are many, many examples of catalytic and other kinds of functional RNAs. So it overturned the idea that proteins are the functional molecules or the sole functional molecules of biology, and opened up the idea that life could have originated in an RNA world. And so this is a very simple world compared to our present central dogma. RNA makes RNA makes RNA, so it carries the information for self-replication and information storage, and then by transcription of small pieces of RNA off an RNA genome, you create ribozymes or functional RNAs that are the equivalent of our proteins. But where did proteins come from? Well, back in the early 50s, Stanley Miller showed that if you cooked up simple compounds like methane, ammonia, and so on with water in a reducing environment and sparking and trying to create the early terrestrial environment, he got more than 11 amino acids, including many that are actually found in our proteins. And later, people analyzed his experiment and his actual stuff that he made and found there were actually over 20 amino acids in there if you looked hard enough. So this inspired a lot of speculation about, there we go, that maybe life could have started spontaneously from protein. But how do you get from the protein back to a gene? So, and the bottom line there is you can't make a gene from its protein. And so a possible solution is that you evolve protein synthesis from an RNA world. And the problem, and that's consistent with the observation that protein synthesis or translation, shown in red here, is very RNA rich. You have, of course, messenger RNA, but you also have transfer RNA, ribosomal RNA, and other RNAs involved in protein synthesis. But why would you do this? I mean, what was the driving force for the evolution of proteins from an RNA world? Well, we certainly didn't have engineers sitting there saying, wouldn't it be great if we made polymerized amino acids 100 or 200 long, and they would fold up automatically into precise three-dimensional structures, and they would even be catalytic. The chances of that happening are I think Carl Woese once estimated an envelope calculation of something like 10 to the minus 80th. So why would you start making proteins? Well, one possible solution is that you started by making small oligopeptides. And these oligopeptides would be made to help RNA, or be selected for by binding to RNA. And one important reason for this comes, or justification for this comes from the observation of what happens when you bind oligopeptides to RNAs, as shown by the HIV-TAT-TAR interaction. So TAT is a protein that binds to the HIV RNA called TAR, and dramatically rearranges the local structure of the RNA, as happens, of course, in many protein-RNA interactions. But Puglisi et al. Also showed that a 9-amino acid peptide could induce the same conformational change. In fact, at high concentrations, a single amino acid, an arginine, could do that, as shown here. So on the left is the naked RNA, TAR RNA, and on the right is what happens when TAR binds to it, or even the 9-amino acid peptide, or even the single arginine shown in orange there. So the idea here is that by having a peptide bind to the RNA increases the structure space of RNAs. In an RNA world, it's great. You have all these catalytic RNAs, but RNA is inherently more or less versatile in its ability to fold than proteins. And when you bind a peptide to it, it increases the possibilities for RNA folds, and therefore RNA structure, and therefore RNA function. And so that could have been a driving force for the evolution of peptide synthesis. So another major problem, very major, is evolution of the ribosome. Ribosomes themselves contain over 50 proteins. So how do you make the first ribosome if you needed protein to make the ribosome? And you didn't have God, probably, but you had something close, Francis Crick. So Francis, in 1968, hypothesized, it is tempting to wonder if the primitive ribosome could have been made entirely of RNA. So fast forward a few decades. So here's the ribosome, the parts list for the ribosome. So it's 2.5 million Daltons. It's a huge, one of the biggest biological structures. It contains 2/3 RNA, 1/3 protein in its simplest form, over 50 different proteins. And it's a very complex structure, as you can see. So where do you start? Oh, there it is. So back to Francis Crick's question, could the primitive ribosome have been made purely of RNA? Ribosomes, stripped of nearly all their proteins, can still catalyze peptide bond formation, as we discovered a couple of decades ago. And decades of studies on the ribosome show that all of its functions are based on its ribosomal RNA. Sometimes the proteins are helping out a little bit, but it's basically an RNA-based machine. And ribosomal RNA contains the most highly conserved nucleotide sequences in all of biology. So, as usual, Francis was probably correct. But ribosomal RNA is itself huge. This is just one of the ribosomal RNAs, the 23S RNA from the large ribosomal subunit, with the secondary structures shown on the left and the 3D structure shown on the right. So this is already huge. Where do you start? There are three core mechanisms of protein synthesis: peptide bond formation, codon-anticodon recognition, and translocation. So if we start with the first one, peptide bond formation, the peptidyltransferase center is actually a tiny, relatively speaking, three-dimensional structure that catalyzes peptide bond formation inside that little circle shown there. So could the ribosome have evolved beginning with peptidyltransferase? So the peptidyltransferase is contained in one of the 6 domains, domain 5 of the 23S RNA. The domains of 23S RNA are connected to each other by what the Stites group called A minor interactions, a single adenosine donor binding to the minor groove of a RNA helix, double helix acceptor. So Bokoff and Steinberg made a remarkable discovery showing a hierarchical relationship between domain 5 and the other domains of 23S RNA. And that's shown here. In red are the acceptor helices, and in yellow, the yellow dots show donor adenosines. And you see in domain 5, which is on the far right, that nearly all of the interactions are made by donors from domains 1, 2, 3, 4, and 6 to helices, helical acceptors in domain 5. So that very strongly suggests that you started with domain 5 and you added the other domains to it over molecular evolution. So it seems reasonable that evolution of the ribosome began with the catalytic domain. Another, let's see, how much time do I have? Okay. Well, you only need 3 rows of the periodic table to make life. So another basic function is codon-anticodon recognition. So how did you evolve the decoding site of the ribosome? Well, the job of decoding is just to enforce Watson-Crick pairing between 2 3-base pair RNA sequences. And this is done in the ribosome by the flipping out of 3 nucleotides in 16S RNA, A1492, A1493, and G530. And in the top, you see the vacant decoding site, and in the bottom, in yellow, is the tRNA anticodon loop, and in blue is the messenger, and you can see all 3 bases flip in to the minor groove of the codon-anticodon helix. And this shows the A minor interactions between between the ribosomal RNA and the message in tRNA. So how did the decoding site evolve from an RNA world? Well, the most likely reason you would want base pairing enforced in an RNA world is in RNA replication. So here's a proposal. So now we go from pretty solid reality into hypothetical fantasy, which I'll leave you with. And that is a proposal for something that we call duplicator RNA. So first of all, it looks kind of like the anticodon stem loop of a tRNA, except that it has a self-complementary tetramer tail. And so it can make a homodimer by pairing of its self-complementary tail to itself. And now the way it works is to read 3 base pairs at a time off a template and bring in random trimer templates until it finds the right one with the decoding site. And so there are exactly 16 self-complementary tetramers possible. And so this could have been the founding 16 tRNAs of the genetic code. And 2 important mechanistic advantages here are there's no double-stranded RNA at the end after you've duplicated because the triplets just fall, or the dRNAs just fall off. And recruiting of oligomers instead of monomers, as you have in RNA polymerases, means that the substrates would bind somewhat stably during polymerization. And you would just have to ligate them together. And there have been many examples of people evolving RNA-based ligases in the laboratory using CELEX. So we think it's not crazy. There are problems, of course, and I'll gloss over those, of course, and leave you with the conclusions that evolution, well, all of the basic functions of the ribosome are based on RNA. The ribosome must have evolved from an RNA world, but how and why? Evolution of the ribosome likely began with peptidyltransferase. RNA binding peptides can bring with them the ability to change the structures of RNAs and enlarge the structure space and functional space of RNAs. And finally, the decoding function may have evolved from an RNA world polymerase. Thank you.